a much higher pace, I mean, they need to do the catch up, as I told yesterday. And uh, the second speaker of the day today is our Oscar Hennings. He is not new to shrimp breeding, though he's working for a new company called Benchmark is an old company, but they're into shrimp breeding for the first time. They've taken over a company in Colombia, and they started their commercial operation. And Oscar Hennings, everybody knows, I mean, he has been working in shrimp breeding for the last 25 years. He was there in Oceanic Institute, where uh, Viban worked. Then he worked for Conabe, and he was with Shimp Improvement Systems as well. And now he is with Benchmark Holdings. In fact, he was one of the first few guys who has come to India to make hatcheries aware about Vaname operation. He was there in India in 2008 along with Matt Briggs, and they organized a couple of meetings. I vividly remember those meetings. I mean, almost 16 plus 6, about 20 years have gone by. I mean, if not 16 years have gone by, but still he's looking young. He's still the same man.
I don't know about that.
So over to you, Oscar. And his talk for today is strategies for dynamic and diverse challenges. Definitely our environment is very, very diverse, and it is dynamic as well, because it keeps changing according to the season in India, the weather. So definitely we need to have strategies for the dynamic and diverse challenges. Over to you, Oscar.
Thank you very much. Thank you very much, Javi. Yes, I still remember vividly that day, that first met. And I remember vividly that the dinner that you offered us was really good. Thank you very much for the introductions. I'm really happy to see familiar faces, everyone here. So I'm gonna start sharing my screen. Let me put a pointer. Okay. So yes, as Avi mentioned, the title of the presentation is Strategies for Dynamic and Diverse Challenges. So before we go into deep on my talk today, I'd like to give a quick introduction of Benchmark Holdings. It's an aquaculture biotechnology company with 3 major divisions: genetics, advanced nutrition, and animal health. Advanced nutrition is a key part of a Benchmark, and is, as you all know, INVE is our advanced nutrition part of the Benchmark. So with the products that it's quite well known in the industry in India as well. On the animal health part, it's mainly for finfish, salmon, but also for sea bass, sea bream, and also for tilapia with vaccines, not as well known in India. That's one of the reasons why I like to bring this up to you. It's one of the 3 major divisions that we have at Benchmark. So now going into my Kuliana, I can say my part of the Benchmark genetics, the Benchmark part, the part of genetics, and the part of genetics in shrimp. But before, a quick introduction also to the Benchmark genetics part. So 40%, just to put in perspective, 40% of all Atlantic salmon farmed today has 40% says Benchmark genetics hands-on, either on our 3 breeding programs that we have in Norway, Iceland, and Chile, but also through partners and also through service provider breeding programs that we provide service for. So here, where are we on our Benchmark genetics, the infrastructure? Besides salmon, 2 sites in Norway, 4 sites in Iceland, 2 sites in Chile. We have a breeding program for tilapia, Nile tilapia on Homestead, which is south of Miami. And we have the 3 sites for shrimp, our Nucleus Breeding Center in Colombia, our Elite Multiplier in Florida, and also our BMC in Thailand. And yes, as Ben mentioned, yes, we have the thinking for a BMC in India as well. So learning from the salmon industry, we saw early on that shrimp has this diverse culture method. When I started the Oceanic Institute, working for the Oceanic Institute in 1999, I could swear, I could bet all my money that by now, 20-something years later, that would be a standardization in the shrimp industry, meaning ponds will have a magical size that works better, a magical size of a hatchery. All those components have been standardized by now, but they have not. I still think that that's going to happen in the future, but I stopped guessing when that's going to happen. But until that happens, there is a need for different products, different lines, breeding lines for different environments. So just a little bit introduction on the products that we do for the different species. For salmon, we have the eye eggs or the ova. For shrimp, broodstock, a small PL market just close to where we are in Florida and where we are in Colombia also. Tilapia, broodstock, but also sex reversed fingerling, again, for the local market. As you can see here on your left-hand side, you have 5 different products or 5 different lines for our genetics in the salmon industry. And we have currently 3 different lines for our breeding lines for shrimp, and we are adding one more pretty soon. So what are those lines? We have, as Ben mentioned on that table, a balance line, which is a select for growth with high survival. So keeping in mind this condition, so it is the line that adapts better to changing environments, to different conditions. And that's the line, the first line that we have in India right now. Because of this, because we need to understand where it performs well, where it lacks something, so where we need to improve or maybe bring a different line. And yes, a different line can be brought in for different areas in India, which is a diverse country as far as environment, but also different times of the year in India. We do have a specialized growth line for low salinity. So it acts on, again, as a resistant line, a more robust line. And our resistant line, BMK Protect, which is selected for white spot and EMS. White spot using genomic tools, and EMS starting to looking forward to start using the same genomic tools as well. But the reason why we are able to develop those lines, those different lines, is because we have a broad genetic base. One thing that Ben mentioned that I agree 100%, coming out of Hawaii, that everything is really expensive here. Space is really limited. It's expensive. So keeping up a founder, a broad genetic base, it's quite pricey, it's quite expensive. On the other hand, in Colombia, where we have our nucleus breeding center, land it's way more available, also as people. And Benchmark Genetics Norway, old benchmark Aquaforce Genetics, had kept the founder gene pool quite intact. And that's for us a main gift, that from that founder population, we can spin out different lines for what is happening today, but also breeding for the future, whatever standardization will come in the industry, the new norms, the new environmental changes, and all that. We kept and we will keep this gene pool pretty much intact on the side for us to be able to spin out the breeding lines that we need, the industry needs. So Again, Benchmark Genetics Norway is pioneer in using genomics in salmon breeding. And that's what we'll be doing. We'll be taking what we learned from the salmon, the technologies, and applying that to shrimp, tilapia, and other major farm species. So going into the genomics for shrimp, the first that we start using as a DNA tool was a parental assignment chip, but we developed in 2018 a custom SNP array together with Neogen, and then later on we improved that to 40K to be able to sequence the whole genome. Now, example of how we use our DNA parental assignment tool. So we went from having us and the whole industry having to tag the shrimp at 1.5, 2 grams, physically tag one by one, to have a DNA panel that we can quite precisely determine who are the families, who are the pedigree of that animal. And that is really powerful. Here, the shrimp goes from nuclei to 1.5 grams, let's say 40 days, 60 days, depending on the growth rate. So that means that those families are by themselves until that time. With the DNA tool, you can mix all the families at nuclei stage. And just to give you perspective, our last batch, we had 9 animals. After we harvest, we clip the pleopods of everyone, I tag everyone, weigh everyone, put them aside, send the samples to be DNA to be analyzed. And we got back and there were 9 animals that belonged to female A. But with male B that never spawned, at least on the record, had never spawned, we performed AI with the male B, but it didn't spawn. We scraped that sperm off. We sent it again with male C, and the female finally spawned. So that proved that a female had a progeny with 2 different males. So 50 with the fresh inseminated one, male C, but there were 9 animals at the end of the trial period from male B. You would never have caught that with a elastomer or any other means. The precision is there, and it's a tool that we have to fully use. Now, going on genomic selections, especially for traits like white spot resistant and other traits as well. Like Jim showed, for TSV, the family selection, the SPF concept worked great. Everyone that were working on disease resistance for TSV was able to move forward quite fast. But for white spot, that was a different story. The heritability wasn't there. You were doing this and you get nowhere. So by using genomic selection, you can get the best families based on the challenge test, but using the genomic, you can get the best individuals from the best families. And that increases your selection differentials and reduce accumulation of inbreeding, because now you know what kind of relationship they have, and increase accuracy of the prediction. So I'll go fast on this, just to explain what we do on a regular basis for our white spot resistant line. We use them every generation, a training population. We challenge them. We understand what the genes bring to the table, each one. And then we had our breeding candidates that are capped as SPF. We see the relationships between them. We breed them, and with that, we do a challenge test again on G1, on generation 1, F1, and the ones that were kept aside, the cohorts, the siblings that were kept aside on the SPF location are our dissemination line for the white spot resistant. So here is just one of the the challenge test that we've done is to show at this time, usually we do this, of course, we pick up the high genetic, the high GEBVs, estimate breeding values. We pick the high ones. But for this trial, we pick the high ones, we pick a random, and we pick a low GBVs, just to reinforce all that population. The population are resistant to white spot. This is coming from a population that is resistance to white spot. So you can see that by using the high GBVs, you're able to jump. And this is, again, don't get the numbers as low, because those are pure white spot challenge trials. And what are the next steps for us in genomics? Here is 2022, up to between 19 and 2022, the performance of our 3 lines. And you can see the more robust the line is, the less growth, the growth rate is lower. And we are using the genomic tools to break this negative correlation between white spot, resistant, and growth rate. Now going a little bit more in other tools that we use. Again, bringing from the salmon, the technologies. We, Benchmark Genetics, had developed breed control database software that was developed for salmon, but it was restructured and adapted for the shrimp industry. Right now, we are using the maturation module. We will have the larvae module and the farm module as well. So up to now, we have over 50,000 data points with different KPIs for maturation performance. And we can track performance and compare over time per country, per client, per batch, per line, protocols, ablation, and no ablation. And why do we want this? Because we want to understand how to move forward, how to develop a better breed, but also to have traceability and understand if there was any batch that wasn't performing as it should be, so we can trace back and see which countries we send that batch to, how are they performing in other countries, other clients, and understand if it was a genetic, a batch problem, or what it was. So we have full traceability of that. And again, by including the larvae module and also the farm module, we can bring that from farm to table, if you will. So one example of what we pick up is that the main KPI is average NALPI per female per month, meaning how much you buy one breeder, one pair, how much they produce per month. So we have example here, country A and country B, with very different production output and same genetics, client of ours. So animals that came out from the same tank sometime, and we see this huge disparity. So we went deeper with the KPIs from the breed control using Power BI. So this is the main KPI, average NALPI per country, per month, per female per month. So we went deep in mating rate and also in the average NALPI per female. And we saw that, yes, country A had a better mating ratio, but not that much difference. The main difference was on the NALPI, the average NALPI per female per spawn. And we saw that biggest chunk. With that, helping the client with a different management and also with a better nutrition or filling the gaps, what you can find in one country as far as fresh feed is not the same as you can find in a different country. So by knowing where the deficiencies are, we can supplement with what is lacking. So again, going to one subject that's really close to my heart, it's non-ablation. So just to state, all Benchmark Genetics shrimp sites are non-ablation. We perform non-ablation. All Benchmark Genetics shrimp lines are selected for non-ablation performance. And quite happy that to see that market perception is changing and non-ablation is gaining traction. PL performance seems to be improved by non-ablation the female. So again, we went back to our breed control to understand, okay, how can we help? Where are the gaps? So we got, again, average now PI per female per month. Now I'm comparing the same country to have a fair comparison and try to use as much as possible the same batch of breeders. And you can see that now PI per female per country, sorry, for ablation compared to non-ablation, it's 23% higher. I see these numbers going up to 30% higher for ablation compared to non-ablation, but I believe this numbers and this logic of comparison is valid here. So looking at the other KPIs, average of now PI per female per spawn, ablation compared to non-ablation, yes, there's a 5% higher. To be honest, I didn't run the statistics on this, but it might not even be significant. But now ablation compare or non-ablation for another KPI, meaning source females, meaning how many mature females in percentage you have per day in your female tank. And we saw that was 19% for ablation and 16, almost 17 for non-ablation. So it's a 14% higher. So we focus on this, and with that, meaning you have to make the female, the maturation interval, you have to reduce the maturation interval for non-ablation animal through selection, but also through nutrition, and even simple as change the sex ratio, increasing the number of females in your tank, in your population, to compensate for now, before the genetics and nutrition catch up, compensate for now for this 16, 17% to bring this up to the same level as ablation females. Now, farm performance, again, the line that we chosen to start selling, to start shipping to India was our BMK yield, which is a balanced line, as Jim Wyban pointed out. And the name is that, is because it gives you the highest yield as possible. So this is compare data from Indonesia from April 2021 to February 22, roughly about 130 something ponds on this. And you can see it has a higher yield. But I like to point out to the coefficient of variation. We use two key points, ADG and also survival. And you can see the coefficient of variation for the ADG is the lowest. Again, the same competitors, the same data set. And the same thing for survival. So that has been the goal, and that was the idea also behind the different lines, is to give predictability, stability to the production at the farm level, at the hatchery level, but we all know that the final client is the hatchery, so the farm. So we need to give them a product that can have a predictability for them, and also that works really well for the processing plants as well. So the last slide that I have is what's the future that we are doing. This is an ongoing challenge test for EHP and also white feces disease. We are on day 28. A couple of days ago, when I prepared this slide, we sent 3 different lines for a negative control that didn't receive any either EHP or white feces. And you can see that 2 of our lines after the white feces went quite low, but one of the lines are right up there with the negative control. We see EHP and white feces as a major problem in India and elsewhere as well. And we want to be ahead and to to tackle the product together with the industry and be able to offer a product that is resistant for those diseases. And why did we choose to do both diseases on the same challenge test? Is that we were mimicking and we are mimicking what happens in the pond, what is perceived to happen in the pond. That EHP open or the pathway for white feces to come in and kill the shrimp. With that, I'd like to thank you all for your attention. Thank you very much for the opportunity, Ravi, to be able to be here today, and looking forward to see the other presentations.
Thank you, Oscar. It was a wonderful talk. We're glad to know that you're working on EHP resistance, EHP along with white feces. They come together. So it's good to know that, you're working on that. And the other thing, what I'm impressed with is you're working on coefficient of variation, and, you are working for, more uniform animals and all that. And your target is not just the hatcheries. You are working at the farm results also. That's the way it should go. Most of the breeding companies seem to be working on those lines, but we're very happy to understand that you're working on EHP resistant lines. That is the one that's going to transform the industry. Today's problem in India and most of the other Asian countries, if you see, it's EHP. That's the major